Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. We'd like to, first of all, take a moment and thank our GPCR Ecosystem partners, namely Domir Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. We hope you were able to join us at the Dr. GPCR Symposium on GPCR activation and signaling. If you have not, you can go back and watch the recorded talks with your premium membership. Also, mark your calendars for the upcoming symposia on July 21st on structural and molecular insights into GPCR activation. If you'd like to be considered as a speaker or have a speaker suggestion, please email us at hello@drgpcr.com. For more information and an updated schedule on all our activities, the easiest way is for you to go and use the links in the footer. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have with me Dr. Pierre Eftekhari. He is my guest this morning. Pierre, welcome.
Thank you. Thank you, Yamina. Thank you for having you here.
So excited to have you on today. And before we hit record, we were talking about making a choice between science and art. So I'm very excited to have you on and talking about your career choices. So why don't we start with who you are and what do you do today?
Right. Well, I would say that I'm now the CEO, founder and CEO of Innovium Scientific that I started 11 years ago. And also I'm CEO of another company, which is called PIMS Technology, and also a third company that I haven't started, but just acquired, which is the BCL Design. In all of three companies, I'm not the real entrepreneur. I'm the scientist. So, but anyhow, it's important to be involved in this. As a background, I actually started working in the corridor of the School of Medicine back in Gothenburg in Sweden. And very early stage, I realized that it's not my cup of tea, really. I needed to have a lab bench and I needed to go more into detail. And to be honest with you, I found medicine very boring. It's not challenging enough. So I actually stayed in the medical world, shifted to clinical physiology, and which I completed afterwards with the mammalian physiology and clinical immunology before starting the PhD here in France, in Strasbourg. That was very much based on the physiology of the 5-HT4 receptor, which is the GPCR.
Hence the connection to the Dr. GPCR podcast.
Exactly.
So, did you always know that you wanted to be a scientist? We spoke about it briefly before we hit recording.
Well, no, I'm was very much actually in my youth very much interested in art, and I did a couple of I would say school of arts change all the time. But something happened when I was 15 years old, and there I became very much interested in science, and I said, finally, I've got two choices, and it was that ICA. A neighbor's mom died of a heart attack, and I saw that kids were going on. Yeah, that was the thing. Nothing more than that. I said, probably something to be done here then. So I used actually afterwards all my knowledge about what I wanted to do, shared between the, I would say, poetry, art, and also science. And finally, as many of us, I think parents will decide which way you should go, and I listened to them, and I chose science. Then, yeah, medical school, but I didn't finish. They just shifted. That was a good bargain for them. That just let me do what I wanted to do. So, did the science, yeah.
So interesting. You mentioned, medical school. Was medical school your first choice, or was it kind of more of a family decision?
No. It was my own my choice, because I was actually thinking that you could, through medicine, you could save lives. Actually, you could do that. There is no problem. But the thing is that when you get to the medical school, we see that is very restricted. So
Yeah.
you need to have something broader. You want to be at the initiation of the, I would say, thoughts and methods and treatments and the drugs and whatever goes with it. And as a medical doctor, you wait until somebody's bringing you the solution, and you apply that afterwards. So it wasn't fun for me, no, no.
I can understand the sentiment when you're a trained scientist or a scientist at heart. You want to be able to find the solution, and you want to be able to understand why is this the solution, and then you ask the question, is there a better solution?
Exactly, yeah. It's the Espiral that actually opens, it's always upwards and bigger, yeah. When you answer a question, yeah, then you've got another question comes out that actually requires more understanding and knowledge, and that's it. And you continue all the time.
Yeah. And you mentioned the 5H2 receptor. When and how did you first hear about GPCRs?
And there was a 5H4 receptor. When I heard for the first time, that was when I was actually an undergraduate in the basic pharmacology, that was looking at the receptors. And that was the first time I came across with the GPCR. That actually at the time I didn't understand very much about it. But the real one came when I moved from medical school to the lab and came across the adrenergic receptors in the field of autoimmune diseases. At that time, late 90s, was very much in thing to have a look at the autoantibodies in cardiomyopathy, for example, hypertension, and looking at the adrenergic receptors related to the antibodies. You've certainly heard about idiopathic myocardial diseases that one would have related to antibodies from the adrenergic receptor 1. That was actually a period that I started to work, and I found it fascinating how we could, through the O2 antibodies, get to the probably pharmacological ligands and solutions. Yeah.
So interesting. I want to go back a little bit because you mentioned medical school and then switching to the bench. Once you realized that medical school was not for you, how did you decide, or what made you decide to go for the bench and work in the lab? Have you had a previous experience that you liked and you said, oh, I think I prefer working in the lab?
I think I always, since you did art yourself as well, seen you manually, and you want to use your hands all the time. So it was a thing that was missing. And when we start to do, I remember very well when we started to do neurophysiology, and then we had the lab work, and then you could really just get involved with the electrophysiology. There, I say that this is the thing that I really want to. And back at high school, I was very good at physics and things, not theoretically. I was very good when we were at the lab, so I could understand things very fast. So I think then I see that, and still I believe today that everything, the word actually is at the bench. There you're observing things, and you can explore things. And if you take distance from the bench, then you've got a bit of difficulties. Serendipity all of a sudden can disappear,
Definitely. And I think I think reflecting on my time at the bench, it was ecstatic. You get into that and you know what you have, you plan it out and you just do it, and it's such an ecstatic experience. It's like you're high on drugs.
Yeah, your heart rate,
yeah. And then you're working on something, and then once you get the data, you start analyzing, and if no matter what, you went through a whole process, and it's the experience of that process that carries you on to the next experiment.
Absolutely, yeah, absolutely. People that are actually, well, that's a good thing that we're sharing the same thing, because I think every time when you're at the bench, I'm now 55 years old, so still I'm doing bench every day. I couldn't imagine my life not being at the bench a single day.
Wow, that is really, really amazing to hear. Let me ask you this. CEO of 3 companies, 11 years in the bank when it comes to building company, and you're still at the bench. How do you do it?
Well, I don't sleep much. I think I belong to these type of people that they need to have for 5 hours good sleep, and that's it. So that's it. And then it is about how you can organize yourself. And I'm very happy. I was a very privileged person having a wonderful team around. So I don't need to be beside and with them, just people actually. And we do have these type of horizontal management here at all 3 companies, that everybody's responsible. When it comes to the signature, it's me, but otherwise, everything we're at the same level. There is not a CEO or whoever it is. So, and that is very good. So you could delegate a lot of things to them. So,
Yeah.
to be honest, I just sign when they say, tell me sign here, and I do. So,
And it's very unique to have the CEO, even if it's a title, and sometimes people make a big deal out of it. CEOs are still human. They still do need to go to sleep and
yeah.
eat. But I think it's still amazing that, you still have this drive to be working at the bench, and also it's unique to have a company, or at least it's more on the unique side to have a company where you can have this flat structure.
Yeah, you need to have talented people around, and I think one of the things that I was, if I say that, successful in, just gathering people around the same cause. So uh, for me, and I think for you, the same thing. The most important thing, what you do, is why you're doing it,
Yeah.
and then it comes to how and what. But why, and I've been able to gather people around that everybody's actually very much interested in why. So it actually naturally brings in very fast the dialogue, and you don't find yourself in 2 parallel monologues. You think that you're in dialogue, but you're not. But with the collaborators, even with the engineers and technicians, they do have right to say what they want, and they do have right to say that they don't want to do it, as long as they can explain it. So we get very fast to the dialogue. So I think. Yeah, the one thing I'd manage to do, just bringing these people together.
Let's you use the correct keywords, the why, the what, and the how, which I think are very important things, and not a lot of companies think about that, about their why. So let me ask you this: what is your why?
Yeah, well, the most important thing for me in science is that, at least in the medical sciences, because science in general, I'm not very sure that it's doing much good, but medical sciences, it is that to put a smile on people's faces when you can contribute, that you take the suffering away from
Yeah.
someone. So that's the ultimate. If as long as we haven't got there, although you develop a lot of tools, technologies, companies, there is no success. That's the thing.
Yeah.
So that's to me, that's the only why is this, and this is medical science. It should serve this,
Agreed.
nothing else. Yeah, yeah,
Agreed. On our end, or at least for me, it's to improve human health.
yeah.
And it pretty much goes to what you were mentioning. And we do this through Dr. GPCR, through bringing people together, through allowing people to meet, to exchange ideas, and to hopefully collaborate, with the hope that this virtual community goes back into the lab, into the different businesses, and then come up with medications to improve human health, which I think is, and it took me a long time to get to this, to figure out, okay,
Yeah.
what's my why, and how can I translate it or make it a reality.
But in the US, I think that I think in the and nevertheless in the Anglo-Saxon culture, you are very much dependent to that why. Nevertheless, that why, let's say that in a liberal society, is defined very much economically, but at least it's there. And there is no problem to say that I want to be rich. I do this because I want to have money. That's it. At least it's clear. But when you get to the more the European, I would say, and or when you get to the cultural languages, the more implicit rather than being explicit. And then these whys are very much obscure. You don't know why. And then, of course, your family will come in and give you a lot of directions that you probably don't want to do, and you waste your time. So no, I do believe that we should always, whatever we do, fine. We're working on a project that is called TRACE, and it's the art, science, and literature that is mixed up. And that TRACE is actually based on that. Whatever we do have a consequence, and that every decision is made will have branches out and will will have effect on our near neighbors and also outside, both locally and within time. And this is exactly the same thing, that science cannot be just related or reduced to economy, although economy is a very big part of it. If you don't have money, you cannot you cannot make, especially in biotechnology, it's very expensive.
Yeah, no, agreed. I think economy is important, but the high level goal should not be to become rich or to make money. It should be to improve human health when it comes to biotechnology and companies. And yes, the money is the enabler, basically.
Absolutely. In that case, maybe you yourself, and also since you're networking at the same time, is that we are building up an institute of personalized medicine that is international, and this is all based on not talking about individualized medicine that is based on the genomic. It's very easy to get there, but the relevance is very hard and very difficult to have any, I would say, causality. We're getting most into the correlations and any type of indication that's a waste of time. And this one actually is very much based on the clinical understanding about a disease and based on the personalized one, taking distance from all the normal values that we are using. So which means that we're much concentrated on what is the individual normal value. And also it's based on another thing, is that in medicine, we're talking about any type of changes and also any type of distance from the normal value is considered automatically as abnormal, which is define a disease. But it is not true that at the individual level it's an abnormal. It should be taken to So that's it's very much based on the approach, first of all, and also a lot of technologies that are coming in.
I absolutely agree. You go to the doctors, you get your blood drawn, and then you get your results back. And if it's 0.02 difference from the quote unquote normal value, it's considered to be abnormal. But they don't take into account the variation between different people. And let's not get started talking about the effect of the environment, the effect of exercise, for example, the types of foods that you consume, and your genetic background as well. And that's unfortunate.
Yeah, of course. Yeah. And above all, what's the most important thing, and one of the things that I was criticizing today's medicine, despite all the money and a lot of effort that all our peers are doing in these things, we still in no man's land, is the fact that we are in the symptomatic treatment still.
Yes.
And which means that we don't have any clue about the pathophysiology. Or if we have any clue about the pathophysiology, it's very descriptive. At the mechanistic level, we don't have any clue. And this is the biggest part of, I would say, the flows that we've got in medicine. Although it's not that the doctors, they don't want to do it, but it is our side, the science side, to bring these to them and work with them all the time. Unfortunately, it's not the case for the time being, but we are hoping with the institute like this, being able to bring that, given the also the role, the conductor role to physicians. Yeah.
I think I think you're spot on when it comes to this. We don't understand, and by the time you're diagnosed, it's too late.
Mm-hmm.
And because doctors are trained to recognize those symptoms,
Yeah.
when the results of your testing is completely abnormal, but since we don't know the link from a pathophysiological standpoint, maybe if we knew, we could diagnose this, or we can shift that balance back to normal ahead of time and avoid having to take medications, for example.
Yeah. Yeah, I do agree with you, but by working on this, I realize as well that the biology is complicated.
Yeah.
But it's not just only complicated because you've got a lot of different factors that are around this. It's highly dynamic. That's the bad thing about it. So when you get to the treatment, then you've got the iatrogenic remodeling that it comes into account as well. And that iatrogenic remodeling is very much person dependent, individual dependent. How can we move forward with this and trying to understand? And it actually brings in a bit, we should be talking about GPCRs, but with thinking around the biomarkers that everybody's talking about.
Yeah.
There is no biomarkers actually you could use today except the 3 surrogate markers that everybody have: blood pressure, blood sugar, and also body temperature. You don't have anything else. Whatever you got is all limited to the population that is studied. And if you get out of the population, it doesn't work anymore because of this actually highly dynamic and the differences that we got.
Yeah.
And at the same time, it brings one thing to it, and it also shows why GPCRs are very difficult to work with, is that for the same function, you could have different path and signaling pathways.
Yeah. Yeah, no, I think you're spot on. Before we dive back into GPCRs, you're involved in 3 companies and you're also building out this institute. Where will it be? Can you tell us a little bit more about the stage of
institute?
Yes.
Well, at the same time, we are actually, if anybody is happy to join us, we are at the building up the core people around the both scientific board and also the management. Mm-hmm. So we need to have a lot of people around, and it is very interesting to have at least physicians in any from many different human disease areas there are, and also from different cultures. So what we've got in the group now is very much based on the Germanophonic people, a lot of from Germany, from Switzerland. We do have people from Israel that I'm very keen to continue working with them. Love working with people from Israel, and we have also we've been inviting out people from Japan, physicians from Japan, from Central Asia as well. That are involved, and also, of course, French physicians also that are involved in this, and some pharma companies as observers for the time being, like Roche and Novartis, but they are observing, they are not part of the group for the obvious reasons. Yeah, they're just observing. Yeah.
Wow.
So if you could have, yeah.
That's amazing. And once we stop recording, we're going to let people wonder. I have a couple of ideas when it comes to physician suggestions on this side of the pond, if that's something you're interested in. All right, let's get back to GPCRs. Okay. I ask this from everyone. What is your favorite GPCR?
Well, of course, if you're talking about the families, that's the rhodopsin family. That's the and I think most of the people that are working with GPCRs are rhodopsin driven people, because everything has started with this. But I could say that I'm also very interested in metabotropic, the third family of GPCRs. Nevertheless, in the group, what I have studied and was still working on is the serotonergic system and also adrenergic system and melanocortin receptor. I must say that in this family, what is very interesting for me is the 5-HT4, because 5-HT4 has got a lot of isoforms, and the differences that they are in this group, mostly in the C terminal, and a couple of them you've got in the second extracellular loop, but mainly is that, and it shows very well the diversity of the pharmacology we could have with just the one serotonin as a neurotransmitter or hormone, how want to classify that, but having a diversity of physiological activity, both from the central nervous system in the gastrointestinal tubes and also what we could have in the heart as well. As a physiology. And melanocortin is very interesting because it has got the direct effect from my side on energy expenditure and also food intake, which also is very interesting to have a look. Both of which are known to be GS, but we know today that it doesn't mean anything. It depends on the system that you're studying. So they are very much receptors that are able to talk with other second messenger systems as well. Depends on you.
Of course, of course. So you mentioned not going to medical school, going for a PhD. We just went through the couple of GPCRs that you really liked working on. Can you walk us through from a timeline perspective? When did you encounter, in what context did you encounter working on these GPCRs? I'm really curious about also what happened after your PhD.
Okay, all right. So when I actually came to Strasbourg to do my PhD, I was taken in because I was physiologist, and at that time the lab was working on autoimmune diseases, very much concentrated on systemic autoimmune disease like lupus and Sjogren. And it was Johan Hubeck that took me in. He was himself from Belgium and was involved in GPCRs. Recently, 5-HT4 was sequenced and patented. At that time, we were talking about a short 5-HT4 and a long 5-HT4 receptor. And he wanted to have somebody as a physiologist to be able to work at the pharmacology of the 5-HT4 receptor using immunology. And immunology was that because the lab was in autoimmune diseases. And of course, very much involved in autoantibodies. So I came in and we start to have a look at the receptor activity. And one of the tools we had was the patient's sera, both from the Chagas disease, because we're talking about, and also very much historically because of the cross-reactive antibodies between the adrenergic type 1 receptor, second extracellular loop, and also trypanosoma cruzi protein. And that was a cross-reactive. So we were looking at what kind of autoantibodies from the lupus patient could cross-react with the 5-HT receptor. So we found a cross-reactive, a cross-reactivity between Rho52, which is the ribosomal protein that is actually one of the markers for the lupus. And it happened that Rho52 was involved also in neonatal lupus, which would give the block auriculoventricular block type 1 and type 2. And once we had a look at this, we had a look at there was this sequence similarity between Rho52 and also second extracellular loop of the 5-HT4. And then I come with the idea that, so why not we induce antibodies in mice mother and have a look at what happens with the pups. And all of a sudden we have this, the model worked actually very well. And some of the Jill Byen in US were able to actually to reproduce and have a look at these cross-reactivity. So that was the whole idea how 5-HT4 came in that idea about that we have a 5-HT4 receptor as an effector, but we have got a cross-reactive antibodies that is actually the immunogen is Rho52, but that part of the Rho52, the antibody that is recognized in the paratope, can also interact with the 5-HT4 and block the activity of the 5-HT4 from the moment that the adrenergic receptor in the heart hasn't taken over. That would be the embryonic period. So we could induce, most of the pups actually, they had a B1, but we had also the second degree of the auriculoventricular blocks. So that was the whole idea. Everything started from there. Yeah.
Very, very physiological, yet you made the link between the pharmacology and physiology in that project.
Yeah, we wanted to actually introduce as a model that you could use immunoknockout. We're talking about that, or immunomodulation, maternal immunomodulation model. So we worked on other models as well, but they weren't really good. As long as you were at the GPCRs, it worked very well. From the moment that you would work on the channels and things, it didn't work.
Mm-hmm. And after your PhD, what was next?
So after PhD, of course, I continue working on 5HT4, but well, I had a research position at the public, I would say, at the academy level as a scholar. But at the same time, I was offered to work in a private company and build up a company from the beginning as a startup, using my knowledge in physiology and working very much on developing animal models and developing technologies for pharma companies as a tailor-made. Mm-hmm. So I was working on this for about 10 years as associated. That time the company was called FTD, and then moved to another name with the New Therapeutic. So that was the whole thing was that from the day one to from the beginning until the end, developing new methods, tailor-made methods for pharma companies. And actually, I took the same model to Innovium. With this time, it's not just the methodologies; it are technologies. Yeah.
So that's great. And then how was it at that time? I don't know how it's perceived in Europe, but 10, 15 years ago, and I think even now today, sometimes people tend to think about industry and the private sector as the dark side. Mm-hmm. And what was it the case when you switched from your PhD to go work at the company?
I would say it's a mixture of the thing, because the people from academia think that everybody's working in the industry is very rich, but they don't realize that when you have got your own company, most of the time you're not even paid, at least from the beginning. So it's something else. You know it yourself because you've got your own stuff. And yeah, of course, there's a whole idea that you're rich and that you're in the industry. And well, it actually coincided with something very funny happened, at least here in France. I don't know how much was at that time in Germany or other European countries. Is that all of a sudden all the expertise that you would have from the academia was moving to industry. So at my time, people from industry would come to academia looking for experts, to develop things. And that was completely the other way around, because here they started to copy and running after papers. So all of a sudden you would see that the quality of papers dropped considerably, and still continue doing that. Hasn't changed. And from the sign of actually the application, that the pharma or industry was taking that and creating value. So it became more like I like you and I don't like you. I like you because you could help me to move forward, and I don't like you because you've got a possibility to be able to do that. Some kind of mix of the jealousy and at the same time envying you. But from my side, my company is very small. We're just 5 to 6 people, so it's nothing really. In that case, I didn't have much of the problems in there, but nevertheless, for the 5HT4, that was late 90s, we had this idea that develop specifically antibodies against the GPCRs and use them as a ligand. So we were actually out, and we had developed a couple of antibodies, and at that time we called it also diabodies that could interact between the, for example, muscarinic receptor and also angiotensin receptor with the two different two different paratopes, yeah, you are using the SCV on all these things. And we went to the pharma and explained that, and they said no. Because we cannot have a look at the pharmacodynamics, we cannot organize that, although the idea is very good, but it remains still I would say, research tool and nothing more. And 5 years after, Roche came up with this biologics.
Yeah, I think you were way ahead of your time. You mentioned antibodies targeting GPCRs, and it's you were way ahead of your time. Now, that's what people hear. A lot of people have started working on antibody therapeutics targeting GPCRs, which is
of course, especially in oncology,
yeah, which I think it's phenomenal, and I think you were way ahead of your time. There was not enough technology potentially to evaluate these therapeutics, and now we're at a stage where we can do that.
Absolutely, and I think it's an extremely powerful approach. That's for sure. Whether that it delivers, at least in oncology, we know that it doesn't. The first line is very good, but still, we got a 5-year survival. It's not that good still. And we are working with people in Central Asia, and they don't have access to any biologics. And the funny thing is that when you have a look at a 5-year survival, the prognostical is exactly the same as you would have in the US and Europe. So the question is that do we need to spend so much money to get So this result is very much here.
So after when you started, in what order did you mentioned being involved in 3 different companies, Innovium is one of them. What's the order in which you started getting involved in these, and what made you become an entrepreneur, basically?
I have problems with but the order is actually that would be continue work in academia, and at the side working with the pharma companies, that would the FTD, and afterwards New Therapeutics, and this took about 10 years, I think, altogether. And I had difficulties, I do have still, despite my age, with the, I would say, authorities. I don't like somebody telling me what I should do. Unless we get into to discuss, and I don't believe in any type of compromise with people. It's about a consensus. We understand, we talk about, we make a decision all together, but I will not make any compromises. And I don't do it with my wife, and she doesn't do it with me either, or daughter neither. So it's the idea of always discussing. Yeah. And I had as a CEO, a physician, that we did have a very big gap in terms of understanding each other. So if I was saying something, he would have needed time to at least a couple of months to realize what I wanted to say, and it was at the time was unbearable for me. So I said, finally, well, I will do my solo star, and then I said goodbye to him, and I started Innovium. Because and still I'm keeping it. I don't want anybody above my head just telling me that we're getting any board, that the board has to decide. To me, I just walk out. It's waste of time.
That very much resonates with me as well. And I think when the end goal, you have your why, then all that matters is that consensus, is that common goal that we're working towards. And if anyone or everyone who's in that company or in that group has a valid point, then we get to debate, and then we make the best decision for the company.
Absolutely, yeah.
To move forward, and it's not about ego, it's not about rank. And I think that very much resonates with me. I think that's very important. And I also do feel if anyone wants to restrict my sphere of actions, especially within Dr. GPCR, which doesn't happen, the last word is typically mine, but we do have this debate as to why.
Absolutely. Yeah, and I just coming to under GPCRs, actually, it's not only GPCRs, but GPCRs is very easy to actually to study and put this forward. The difference between the optimizing and making the best of. When you make a best of something, it's very much related to yourself, But when you're optimizing, it's very much related to everybody involved. So
Yeah.
everybody is actually taking it. And biology is doing like this because otherwise you cannot actually survive. So you've got always these optimizations happening.
Yeah.
And if you want to study that, to me, the best one that actually gives you enough tool to study are GPCRs, because channels are too fast.
Mhm.
And if you have a look at the other membrane proteins like the GIPs and glycoproteins that are around, you wouldn't have as many as tools we've got today to study these phenomena. That GPCRs are not working alone. GPCRs are in the heterodimeric form. If it's not the oligomeric forms, you cannot find any GPCR receptor alone, and they are working from different angles with different signaling pathways that could be simultaneously and not sequentially. And it shows very well that the best way to optimize and not making the best of
No,
is
that's
GPCRs to study. Yeah.
No, agreed. And it's interesting that you mentioned heterodimers and protein complexes. I was playing around and I was on ChatGPT the other day, and I asked the question about the top 3 advancements in the GPCR field, either in the last 5 or 10 years. And one of those answers was around protein complexes. So in the context of GPCRs, the other one was bias signaling, and the third one was cryo-EM. Which I think
of course they're carrying,
which I thought was really spot on when it came to technology. But I think you're right. Maybe because we're biased towards GPCRs. I think, thinking about glycoproteins and heparan sulfates, very important proteins, with a lot of sugars on there, but I think they're way more difficult to study. And we don't yet understand that. For GPCRs, we have a basic understanding. We have a basic set of tools. That doesn't mean that we can go home. It just means that we have something to start with. And I think in the past 5 to 10 years, the field has just been propulsed into the future from a tools perspective, but also from a knowledge perspective.
Absolutely. A lot of dogma about like the keyhole pharmacology is gone. And this is actually comes thanks to GPCRs. Otherwise, you'll still be thinking about it. If you didn't get to the key and keyhole to open the door, and also the whole idea about the receptor and effector has changed as well. So we don't talk about as it was completely sterile, saying that you've got a receptor and afterwards the effector. We could understand that it could be interchange as well. The effector could become a receptor and the receptor could become an effector. It depends on where we are standing and how the physiology is working.
No, agreed. So now let's talk a little bit about what you do on a daily basis at Innovium, and what do you guys do? You mentioned developing tools and technologies, but I'm really curious to hear more about the company.
Okay, well, Innovium was based on the Innovium. Actually, the name that I choose is for innovation with one N, and EM at the end is the 2 first letter of Emily, our daughter. So just I wanted to have with her. So the goal of Innovium is just being at the forefront of technologies and developing always, as each scientist would do, new technologies, new methods to overcome the limits that we've got in the medical sciences. So I'm very much concentrated on this. So what we do is as a tailor-made service for pharma companies based on understanding the mode of action or clinical mode of action of a compound at one hand, and also stratify patients as a responder and non-responder. So there are 2 proprietary technologies that are related. The one for the stratification of the patients, the PIMS technology, there's an infrared spectroscopy method is patented, and this is delivered in US, Japan, China, and Europe. And now we've got our second series of machines that are building up. And the goal is that at the end of this year, being able to position these machines in different hospitals and generate data and see how far we could go. And for the target deconvolution as a label-free technologies, in the world there are 3 different companies that are doing the label-free for the time being. It's Pelago, certainly you've heard about. It's in Sweden by Thermal Shift. We've got Diagnosis, yeah, that's in Switzerland. It's a company that is a spin-off from ETH in Zurich. And they are very much based on the biochemistry of proteins. So they do the partial digestion to get there, and ourselves, that we are very much based on the ligand induced pH shift. So 3 different companies in the world are doing that. And our aim is not to understand the primary target. Everybody's talking about primary target because this is the way that we conceive pharmacology. What we are talking about is if there is any primary target, we should know what is its quaternary structure, which means that we need to have the related signaling pathway that is actually describing the observed pharmacology. So that's what we do on the daily basis, and we are agnostics. So we could work in any type of diseases and also in any type of, I would say, ligand, whether that's small molecule, antibodies, peptides, or any type of biosimilars. And also we've got a lot of GPCRs around to work on, mainly based on what I told you, that not the GPCRs on its own, but what are the crosstalks that we could have a look, and that should be clinically relevant. So to do so, and this is actually one of the things that I was talked about later when you asked me about the 3 top wow stuff. We are directly working on human tissue in a pathologic environment, and trying to have a look at the different populations, having a statistical power, and see the relevance of the identified target in a pathological environment, see whether you could do that. So when we're talking about a target deconvolution, it's not just you identify the protein or proteins, you validate their interactions, you validate the interaction of the target and the compound, whether we're talking about, and also that interaction has a pharmacological function. So you go in from the biophysical methods until physiological methods. That's our daily basis.
I love it. And then we can talk about the 3 aha moments that you had as a scientist.
Exactly. Yeah.
But before we go to that, I was listening to you, and it reminded me the beginning of our conversation where you were mentioning that you were all different, and then doctors are trained to recognize a certain set of symptoms, but we don't yet know the connection in the signaling and the pharmacology in the pathophysiological setting. And I think it ties really well to what you were just mentioning, is that your goal is to really better understand the action of a molecule, of a potential drug on the pathophysiological, in a pathophysiological setting, which I think is really important.
Absolutely.
Which is
so that's the whole idea. That is actually why I started in Innovium, because the idea was that we're building up tools, and the way I'm putting Innovium forward is that in just a very small window, if it's not a whole that we've built up with these technologies, we still need to have a lot of people coming in and bringing their knowledge. If we could work economically together to build this up and bring more technologies on the spot, so that is very important to have that. If you have a look at the majority of technologies that we have developed, are all descriptive. Even if we're talking about a cryomacroscopy, electron microscopy, we are observing something, we're describing it, and then we deduce from that. And we very much, as I said before, we are trapped in correlations, and correlations in medicine is the one that actually stops us from moving forward. We need to go to causality more and more. And you would say 10 years ago that we don't have any tools, but there are so many technologies that are available now
Yeah.
The only thing is that we need to have a look at the problem differently.
Exactly. All right, so let's talk about the top 3 aha moments that you had as a scientist.
Yeah, well, as a scientist, well, I think the very first one was that when I saw that we could do manipulate the receptor by antibody. That was that's something. Since I was coming from clinic, I have an understanding of the clinical physiology, and also we're doing immunopharmacology. So, wow, this is a tool. We could use that to develop any type of ligand. Actually, we filed a patent on one of these approaches, and that was the first one, to use antibodies, to use GPCRs as a therapeutic target, and to use antibodies as the ligand. So that, and of course, with everything that I did afterwards was that to show that the second extracellular loop of GPCR, the rhodopsin family, is a very good target to work with. It's interesting to work, and then. The second one came that at that time I was thinking about, like many other people working on GPCR, thinking that we're dealing with a expression of the receptor, and that expression of the receptor is deciding for everything else. And then working on hypertensive patients with the cross-reactive antibodies with 5-HT4, I realized that these patients they would have 5-HT4 as the effector, but they had heterodimerization with the angiotensin 1 receptor. So you would have actually the receptor was 5-HT4, but the effector was by angiotensin 1 receptor. So you would, the autoantibodies from the patient would interact with 5-HT4, but you would have a increase of the hypertension through the angiotensin. And that was the pathophysiology that had, and then you could, when you were working with losartan, for example, when you were working with atropine, when you were using statcholine, all of these things to see whether you could bias that. The only thing that would work, that was a thing that would, the moment that you could block the autoantibodies by any type of serotonin ligand, that you would bring the patient back. So that was another, wow. So we're not talking about homodimers, or we're not talking about the effector receptor. We're talking about a far more complex system, and that complex system should be taken into account in any type of drug development. Parallel to this is that when you have guests around for a wonderful, I would say, a meeting, an evening, it's not because you're a very good cook. It's also because there are people around. It's actually at what time the type of food you've got on the table makes sense. Yeah. But a second after, it's who is eating it, with who you are sharing it, and how things are moving. If you got somebody that actually is put in the face, will destroy the evening, you know. So that's the whole idea. So again, in terms of and that came actually was thinking about GPCRs. We're going to have to have a look at this high ability, and also the rule in all the remodeling, tissue remodeling, that we could have a look. So that was the second hour. And the third one came is that when you study all of these things, you're using models, okay? And then you realize that this is not a clinical reality. It's just based on the models, because you building up these models, you've got the idea. The models are not false. What you study is nothing to through. You're creating still value. You're creating still knowledge, but that knowledge shouldn't be the one that you would use that to transpose afterwards in clinical development and use that as a tool. Then I understood that, oh, there is a difference between individuals. So we don't have the same molecular setups, and we are going from for the same symptom, we might have different underlying pathophysiologies. And that was a third one. So the consequence of that I started in Uvi and said, I don't want to do any models anymore. I want to work with human tissue, with human, and building up this. So that was the whole idea. So that was a 3 aha. Yeah.
I love it. I love it. And then you're right. I think it's whenever you're building a model is great to generate knowledge. Sometimes the model is correct, sometimes it isn't, but then you have that gap between the patient and the model.
Yeah, that's
And
the whole
that's
thing. Yeah. And the model on its own is that you can actually get to a model if you can isolate a knowledge, if you can isolate something. But in human disease and human physiology on its own, it's so complex. How can you isolate that? Whatever model you do, you are partial in it. You cannot
yeah.
go, you cannot do everything. And have a look. There are some of the indications that we have never been able to have any model on. Cancer is one thing. Everybody's using it because there's nothing else to do, but it's a waste of time. Instead of using something that it doesn't bring any knowledge, it's at least clinical knowledge. It's better that you concentrate ourselves to develop something that would have. Um, we have in psychiatry no model actually. All the human psychosis that we do in animal models are just, to my mind, is a waste of time. And that is why I stopped working with animals. And you've got a depression. What is depression? How can we define
Yeah.
depression? Yeah,
Yeah.
and the same, all of that, what you develop things that you lose in your time. Nevertheless, they have actually has produced some knowledge, but I would say that, but FDA is in the same line as they say that there is no use to work with the animal model. Still, we do have enough tools at our hands to be able to develop compounds without going through small laboratory animals and
Yeah.
big ones. Yeah.
No, agreed. You're asking what is depression, and it's such a difficult question to answer. For example, postpartum, you go to the doctor and they have you fill out this form and they ask you questions. And I was just thinking to myself when I was there, I could I could very much be lying and no one would figure out that I'm depressed. Is there any way to give a blood sample or to give a saliva sample, any kind of sample where it's not where you can detect that? What are the changes that the body goes through if somebody is depressed? And can we categorize the phases or the deepness or of the depression through some kind
Yeah.
of a biomarker? Because we rely
Well,
on people's answers.
serotonin history has actually brought a lot of good things in this, but nevertheless, it's a part of that. And is all saying is that the happiness is very difficult to catch. It's almost impossible to find it outside, and very difficult to grasp it inside yourself. So um,
Yeah.
it's not for that. At the molecular level, so many different hypotheses around this. I don't say that these hypotheses are not right, but they are hypotheses very much limited in a very narrow um, path that we've used to study. So most of the time, since almost we don't do any more empirical studies, so everything is mostly induced or deduced.
Yeah. Yeah.
So it makes it very difficult. That is why it's very important that if we want to have anything, we should go to clinic. So it's not from bench to bed; it's from bed to bench and return.
Yeah. No, agreed. Agreed. All right. Last but not actually two last questions. Any advice for young scientists who want to contribute to science in general or to the GPCR field?
The very first one I would say that I say to everybody that actually comes here to work with us is that it is prohibited to use literature.
Okay.
You should think by yourself, have hypotheses by yourself, find the methods by yourself, do it. Once you've done it, then you're going to have a look at the literature. What has happened, where you're standing. Exactly when you are in the middle of the desert, you get out of the desert first, and then you look how the others have done. So that's the thing. The first thing, no literature. Otherwise, you're biased. And especially with the young scientists, we should know that we have enough knowledge to be able to set up an experiment on our based on what we've learned. And at the same time, we can challenge that, because if we use already existing data and we build up. Whatever we do at the best, we just are copying the methods of the others. And the second thing is that it's not that your results are reproducible, that you're not an artifact. So you should be sure if your results are reproducible, the hard part actually starts afterwards, that you're going to have to show that it's not the artifact that you're looking at. So that should, the second thing I would say to all scientists, that everybody's coming to me and I said, I've done the result 3 times, have a look at my CVs, have a look at the STs, and have a look at the population of the CVs lower than 15, to say that, okay, that's very good. I want to see that we are looking at the real function and not an artifact. And believe me, and you know it yourself, majority of what we do is our artifacts, because if they weren't good, with all the money and time and all the brain that is behind all of these things, we would have pushed forward far more better medical sciences that we've done.
Yeah.
So that's I would say and the last one is the most important thing: do things based on credo. If there is no credo, don't do it, because it's so difficult afterwards. You will be so much bounced by all the events that are in front of you that you cannot stand up. So you're going to have to do things by credo. If you think that what you are looking at, or the scientific questions you've got, and this is the one that you believe in it, then it's the thing. Even if you leave it now, you will certainly pick it up a time later in the future, but it will never die. So that's a fair thing. If you don't have any credo, don't start.
I love it. Last but not least, if and when you have job openings at the company, where can people find these?
Normally we do, when we've got things will be out again. The first thing we do is LinkedIn. We use LinkedIn, the first one. Then afterwards, now we know that you exist. We could use you. We use Dr. GPCR. We didn't know it before. And also we could use headhunters if there is a very specific thing. So we could use headhunters. We've been using in the past a couple of times for a specific positions, having headhunters. So we do that as well.
Perfect. And yes, I think everyone should be in the ecosystem and everyone should have a LinkedIn account. Nowadays, if you're not on LinkedIn in general, it's hard for people to find you and find out more about you. I was doing some literature search, and I was trying to find some scientists who authored very interesting papers, and I was not able to find them online, period. Not only on LinkedIn, not even online on the university's site, which I think it's 2023, so people should have some kind of a profile, and the best profile that you can have, that you can take ownership of, is LinkedIn. But then if you're working on GPCRs, you must come to Dr. GPCR and have your own LinkedIn-ish profile on our site.
Absolutely. I will talk to Alexandra about that. We're going to put it on. That was nice to talk to you. That's great. Yeah.
Perfect. Thank you, Pierre. I really enjoyed our chat. Don't go anywhere. We're going to let people wonder what we're talking about after we stop recording. And thanks again for being here today.
Thank you very much. Thank you. Great. Yeah.
Thank you for joining us for this Dr. GPCR podcast episode. We would like to take a moment and thank our guests, as well as our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Please also don't forget to subscribe to the Dr. GPCR monthly newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. And until next time, stay safe.